Welcome to the first lecture in BIO 104, where we will go over an introduction to evolutionary theory. You may ask yourself, how does a population actually evolve? The theory of evolution originated with the idea that natural selection is the mechanism that can cause the evolution of a population. We also now know that there are many different mechanisms that can cause genetic variability within a population. Natural selection is one of them, but we can also have different mechanisms such as genetic drift and gene flow. All of these processes can cause the evolution of a population. If we want to understand these processes, we can use microevolution to examine the actual changes in genetic frequencies or allele frequencies over multiple generations. Understanding genetic diversity is key to understanding evolution. An organism's genotype or genetic makeup is typically reflected in their phenotype or overall morphology. Differences in phenotypic traits usually reflects differences in genes and differences in DNA sequences. Phenotypic differences can be due to various reasons. Perhaps it is a single gene variant, such as a difference between dominant versus recessive gene and how it is expressed. It could be a difference in many genes, such as gradient traits, such as height, weight, etc. And in addition to that, environment can also play a role on how genes are expressed. Remember, natural selection needs variability in order to act. It's considered the basic inheritance of offspring. This contains the genetic information that will influence the phenotype of a trait. A locus is considered the location on a chromosome where a gene is actually located. Within genes, we have two different alleles. An allele is considered a variant form of a gene. Humans, for instance, are diploid, which means that each of our cells contains two different alleles for each locus. We are considered homozygous if we have two identical alleles, or heterozygous if we have two different alleles. Let's say, for instance, I am considering the B gene. I may have inherited a big B from my mom and a big B from my dad. I would be considered homozygous. However, if I inherited a big B from my mom and a little b from my dad, I would be considered heterozygous. Because we have two versions of the allele, we are considered diploid. Haploid, however, refers to having one allele for a locus. Our gametes, for instance, are considered to be haploid. These alleles can be described in different ways, such as dominant, recessive, or codominant. There are different ways in which we can measure genetic variation. We can look at gene variability or the heterozygosity of a population. This is done by measuring the percent of loci that are heterozygous within a population. We can also look at nucleotide variability, the variability in the actual nucleotide sequence. This is done by comparing gene sequences between two or more individuals. However, nucleotide variability, if minor, rarely results in phenotypic differences, particularly when these differences occur in any of the non-coding regions of the DNA, or if these changes do not result in a change in the amino acid sequence, meaning that the phenotype expressed is the same. There are many sources of genetic variation within a population. Genetic variation can result from mutations in the DNA sequence, from gene duplication, from rapid reproduction rates, and also from sexual reproduction processes. We are now going to take a look at each of these processes. A mutation can occur whenever the DNA sequence is changed. Remember, it is only heritable if it actually occurs in the germ cells, meaning that the germ cells will then allow for this mutation to be passed on into the offspring. If a mutation is not heritable, meaning it occurs in any of the autosomal cells, it cannot be acted upon by natural selection. A mutation can occur as a point mutation, which is a change in a single nucleotide. This can occur due to errors in the DNA replication process, or it can also be due to various external factors such as UV light, radiation, or exposure to various mutagens. Some information lead to the of new alleles. Many of these can result in the following. Most of these mutations would be considered harmful. This means that they would be selected against in the population and would not be passed on to the next generation. However, some of these mutations may remain hidden in the form of recessive alleles. In rare instances, the formation of the new allele may be beneficial. This would occur if the formation of the new allele allowed for the expression of a certain phenotype that would increase the fitness of an individual and increase its ability to mate and reproduce and pass on the genetic information to its offspring. However, the formation of a new allele may also be neutral, which means that there is no selective advantage or disadvantage to the individual. This typically occurs when mutations are in the non-coding region, or if it doesn't cause a change in the genetic code due to redundancy, meaning that the third wobble position is not affected by the change in nucleotide, meaning that it codes for the same as no change in the outcome of the expression. There could also be alterations in gene number and gene position. Many of these chromosomal mutations are considered to be harmful. These include deletions, where portions of the DNA is deleted, duplication, where sequences of DNA are duplicated multiple times, or translocations, where a genetic sequence is moved to another location on the chromosome or on other chromosomes. However, in rare instances, alterations in gene number or position can be considered beneficial. Some of these link genes in a positive manner. In addition, some duplications of small portions of DNA can be less harmful and thus they may persist in a population. This provides a mechanism for further mutations. This can result in genes with new functions. If this function causes an advantage for the individual, then it would increase its fitness the ancestral odor detecting genes. Humans have over 380 copies, while mice have 1,200 copies. This indicates that there is an extreme amount of gene duplication that occurred within the evolution of this gene. An important aspect to consider when looking at the genetic variability of the population is the complexity of the genome in combination with how fast those individuals reproduce. In general, mutation rates are quite low in animals and plants, with 1 in every 100,000 genes per generation having a mutation. It is also considered low in many prokaryotic cells. However, because most prokaryotes are very small and can reproduce at a very high rate, they have a very short generation time. This allows for mutations to quickly accumulate within a population. Viruses, for instance, have a very high mutation rate in combination with the short generation time, leading to the rapid evolution of viruses, as we can now see through the mutation rate of COVID-19. Sexual reproduction is a very important aspect to consider when looking at the genetic variation of a population. When undergoing sexual reproduction, there is recombination of alleles, meaning that alleles from the parental gene pool will mix. This can occur during the crossing over phase in prophase 1. It can also occur through the independent assortment of chromosomes. Chromosomes separate independently. Once again, this occurs in prophase 1. And also through the effects of random fertilization. There are many sperm that are produced that are genetically unique, and each one is equally likely to fertilize the resultant egg to form an embryo. This means that there are tons of possibilities for the genetic combination of offspring. The previous slides explore the genetic makeup or genetic variability with respect to an individual. However, there can be genetic variability within the population as a whole. A population, in this instance, is referred to a group of individuals of the same species that breed with each other and produce fertile offspring. Within a population, you will have a gene pool. This describes all alleles for a certain loci that exist within that population. When studying population genetics, it's important to understand the Hardy-Weinberg equation. This equation can be very important and can be used to determine if a population is evolving or if the genetics of that population are changing. We can now use P and Q to represent the allele frequencies. P plus Q should equal 1, meaning that if you have two alleles the frequency of those alleles together should add up to 1. If a population is at equilibrium or Hardy-Weinberg equilibrium, that means that the following equation will hold true. P squared plus 2pq plus q squared equals 1. To give you an idea of the genetics of a population, let's consider the different phenotypic variants of a flower. A red flower has C big R, C big R. A white flower is recessive, would have CW, CW, and a pink flower would be a heterozygote containing the genotype CR-CW. Let's say, for instance, that in our initial parental generation, 80% of our individuals have the dominant or CR allele. This would correspond to P equals 0.8. And let's say 20% of the individuals in our parental generation have the recessive or CW allele. This would mean our Q value was 0.2. If we carry out a Punnett square, as we normally do when understanding genetics, except now we add our probabilities for our alleles into the mix, we would see that CR has a P of 0.8, CW has a Q of 0.2 in the sperm, and we have the same values for the eggs. That means the probability of an individual or offspring having CR-CR would be 0.8 times 0.8 or 0.64. This would represent our p-squared value. If we consider the makeup of our heterozygous individuals, we would have, in one instance, this would be referred to the pink individual on the right, we would have PQ, which would be 0.8 times 0.2 or 0.16. If we take a look at our other pink individual on the lower left, we would also note that it has a value of, or probability of 0.16. This results from the Q value 0.02 from the eggs multiplied by the P equals 0.8 from the CR in the sperm, which would result in 0.16. If we take a look at the last probability for our offspring in the lower right corner, we would have Q squared, or 0.2 times 0.2, which equals 0.04. If we take a look at the genotypes in our offspring, we would have 64% being CR-CR, 32% being CR-CW, and 4% being CW-CW. If we take a look at the probability of our alleles in this offspring generation, we would see that 64% have CR, as well as 16 or half of our heterozygotes have the P allele. That would mean 64% plus 16%, which would equal 80%. If we take a look at what our Q values are in this offspring generation, we would have 4% of our homozygous recessive individuals and plus 16% of our heterozygous individuals. This would lead to 20% of our population having the CW allele. Note that our allele frequencies are the same as what we started with in our include the population is at Hardy equilibrium and no longer evolving. The evolution of the population occurred when alleles are being passed to subsequent generations in different proportions. This can lead to adaptive evolution, where traits that can enhance survival will increase within a population and increase over time. An example of this is studying the resistance to DDT in Drosophila melanogaster, or fruit flies. DDT was a common pesticide that was used in the 1930s. This was widespread and caused the death of many of our important species. With respect to fruit flies, if you examine the allele frequencies of the DDT genes within the population, it existed at relatively low to non-existent numbers, meaning that 0% of the population actually had these resistant alleles. However, after DDT exposure after the 1960s, the genetic analysis of fruit flies demonstrated that more than 30% of the population had these DDT resistant alleles present. This means that they were undergoing adaptive evolution to develop DDT resistance within the population, thus increasing their survival. In order to understand the theory of evolution and how natural selection works, it's important to understand the concept of fitness. Many times you'll hear survival of the fittest, but this term is very misleading. This is because fitness is a concept that will change along with the environment. It's more accurately expressed as relative fitness. The contribution of an individual with respect to their gene pool really depends on the contributions of others in the population. So an individual's fitness is going to be based on how it performs with respect to other individuals within the population. Also keep in mind that the environment is always fluctuating and changing, and so the concept of fitness also has to change. A high fitness level refers to an individual that is highly likely to survive. As a result of this high survival rate, it is more likely to find a mate, reproduce, and pass on those advantageous genes to the offspring. If environmental conditions change, that individual may no longer be the most fit individual and thus would suffer a decrease in survivability and a decrease in the ability to pass on those genes to the next generation. Fitness of an individual is highly dependent upon three main things: survival of the individual, the ability of that individual to find a mate, and the overall reproductive success, meaning that that individual was able to survive, mate, and produce a viable offspring that would then be able to pass on their genes. There are also many different types of selection that can result. Natural selection is not always forward driving, meaning that it is not always pushing in one direction. If we take a look at this diagram, we can see that we have many different phenotypes for fur color. Once again, the phenotypic variation is typically a result of genotypic variation. We can see that the phenotype of fur color has a normal distribution within the population, meaning that most of the individuals will fall in the middle range of color, with very few individuals being on the light extreme of the spectrum and very few individuals being on the darker end of the color spectrum. If we have directional selection, this is when natural selection will be pushing towards one or two of the variants. In this instance, in this diagram, we can see that the directional selection is for the darker variant. This would push the resultant evolving population more towards the darker side of the coloration scale. We can also have what's termed disruptive selection. This is when you would have the decrease in the mean of the original population, and you would see an increase in both ends of the spectrum. In this example, we would have more individuals on the white end of the spectrum and more individuals on the darker end of the spectrum. You can also have what's termed stabilizing selection. This is where you would see a decrease in the variability of the population and an increase around the mean. In this instance, you would have an increase in brown individuals. Sexual selection can also play a very important role in driving the evolution of a population. Individuals with certain inherited characteristics may be more likely to find a mate and reproduce. Many times, sexual selection leads to sexual dimorphism, meaning that there is a distinct difference in the appearance or phenotype of males versus females. If we look at the box on the right, you will see the male individual has the plume of coloration, whereas the female individual tends to be more dull in color. This is very typical for most sexually selected populations, as the females divest most of their energy into egg production and mate selection, whereas the males divert most of their energy into mate attraction. You can have intra and intersexual pressures. Intrasexual pressures refers to individuals that are within the same sex because they will be competing for a mate and competing against each other. However, you can also have intersexual selection. This is when individuals from different sexes compete, and this is very common in populations where you have mate choice. Another mechanism that can cause the evolution of a population is genetic drift. Genetic drift occurs when chance events cause the allele frequencies to change within a population. Genetic drift has predominant effects in small populations. In this instance, you don't have adaptation that leads to an increase in survivability and an increase in the ability of those genes to be passed on to the next generation. So the concept of survival of the fittest does not apply in this instance. Let's take a look at the image below. In generation 1, we can see that our allele frequency is 0.7 for the CR allele and 0.3 for the CW allele. Let's say that 5 of the individuals in that resultant population survive due to some unknown circumstance. If we take these 5 individuals and take a look at their alleles and see what their potential offspring are in generation 2, we could see that in this instance our P and Q values are the same, meaning that the proportion of the CR is equal to the CW allele within our population. Now let's consider that there was some other chance event that eliminated out most of the population. However, it left 2 of the red flowers to produce offspring. In this instance, for our generation 3, we would see that our P frequency, allele frequency, would have increased to 1.0 and our Q would have decreased to 0.0. This is an example of genetic drift, where random chance events have eliminated out certain individuals from creating a subsequent generation, thus influencing the allele frequencies that result from that and causing the overall evolution or change in the allele frequency in a population. There are 2 main ways that genetic drift can cause alterations in the allele frequencies. When the founder effect takes place, this occurs when a small population becomes isolated and serves as a source population. In this instance, you may have a large gene pool of individuals. However, if some of these individuals then become isolated from each other, they may actually serve as a founding population. In this instance, the resulting population and the frequency of the alleles would be highly dependent on those original founding individuals. In the bottleneck effect, this is when a sudden change or disturbance causes a severe reduction in the population size. Thus, the individuals that end up surviving become the founding population. Thus, the genetic allele probabilities in their surviving individuals would then determine what the allele frequencies are in the resultant population. Gene flow can also change the allele frequencies in a population. In this instance, there is a movement of alleles between populations due to the migration of individuals or the movement of gametes. This may result in converging populations into a common gene pool. Typically, this can reduce the genetic variation between populations. For instance, if you take a look at the banding pattern of various water snakes from Ohio to Ontario, there are 2 distinct banding patterns that are present in Ohio and in Ontario. However, in locations where these populations can mix, we can see that there is actually an increase in the variation of the banding pattern that is typical on snakes that are found within the islands between the Ohio and Ontario mainlands. Overall, there are many mechanisms that can cause the evolution of a population. Hopefully at the end of this presentation you could recognize that though natural selection was one of the main recognized mechanisms for the evolution of a population, there are many other mechanisms that can come into play that can change the allele frequencies of a population and cause that population to evolve or change over time.